So, do you believe me when I say that fibrosis contributes to more than 40% of deaths in the industrial world? That is actually true. My name is Sofia Mayans, and I'm the CEO of InfiCure Bio. And the purpose of today's presentation is not to attract investors right now. It's rather to make sure that you guys know what we do and that we exist. So, InfiCure Bio is a preclinical CRO that was funded in 2015 by me and Professor Dan Holmberg. We're a spin-off from the University in Umeå. And when we started the company, we started as a project at Umeå Biotech Incubator. But nowadays, we're actually standing on our own legs with help from our investors, Nordic Light Capital and Partner Invest, as well as with help from some soft money from Vinnova, EU, and Erling Persson Foundation. So, at InfiCure, we are very interested in fibrosis. And as I already told you, fibrosis contributes to more than 40% of deaths in the industrial world. And despite this huge impact it actually has on human health, there are no efficient antifibrotic drugs on the market today. So fibrosis, that's scar tissue formation within an organ. And this scar tissue is formed when the organ is harmed, exactly in the same way as when you get a scar on your skin, you get scar tissue formation. So you can harm your tissues in different ways. You can drink too much alcohol, eat too much fat food, smoke, be exposed to chemicals, or have some sort of disorder. Fibrosis can be formed in any organ, and if enough fibrosis is formed, the organ will completely stop working. We know that most big pharma are developing drugs to treat fibrosis, but something is holding this development back. And one thing is that there is a huge need for new and really good preclinical models where these companies can actually test the effect of their new drugs. And that is exactly where we come in. Since we have developed this NIF mouse, it's a new and unique spontaneous fibrosis model. And this mouse spontaneously develops fibrosis in several organs. It's on the market today as a liver fibrosis model, but as I will tell you later, we are also validating the kidney fibrosis right now. So what we do is that we make drug development more efficient by offering our customers to test the effect of their new antifibrotic drugs in this NIF mouse. So our business model is that we have a fee for service. So what is happening today is that our customers, they send their drugs to us. We treat the animals, we do all the analysis, and we send back the results to them. And it's a fully customer-based setup, so it's always up to the customers how they want us to treat the animals and which analysis they want to see in the end. When our patent is approved, we will start to sell licenses on this mouse. And this means that if you buy a license, you will get the actual mice and you can do all the treatments yourself and all the analysis yourself. And this, of course, makes our business model very scalable. But we aim for becoming a first-hand choice for tests of antifibrotic drugs. So our market then? Well, actually, our market is the entire world. Since our customers are pharmaceutical companies, biotech companies, academic researchers, basically everybody who are developing drugs to treat fibrosis. And just a quick search in global data, which is a market database, has shown us that we have a low estimated market of more than 300 million Swedish crowns only in US and Europe. And then we have a lot of the world to cover besides that. So the reason why I can say that the entire world is a market and that basically everybody who are developing drugs to treat fibrosis are our customers is that we reside in the preclinical segment of drug development. And this means that, when you develop a drug, you have to test the effect of your drug in an animal model before you can go into humans or do the clinical study. So we reside in the preclinical segment. But of course, we are not alone. There are competitors on the market. But when we look at liver fibrosis, common to these competitors is that they are induced. Our NIF mouse is a spontaneous model, but the competitors, they are induced. So you have to harm the organs. You have to harm the liver in some way. You treat them with chemicals or you do surgery, and then they start to develop fibrosis, and you can test your drug. One drawback is that this is not very similar to what you see in the human condition, since most of the time you have a chronic inflammation that is damaging the organ. Another thing is that you get problems with reproducibility. And because you get variances in the levels of fibrosis to start with, so then it's hard to know does my drug really give this effect, or is it just the mouse that had a lower level of fibrosis to start with? And then, of course, it's both time-consuming and labor-intensive to do these inductions. So advantages with our NIF mouse is, as I said, it's a spontaneous model. We don't have to treat the mice in any way. And we see the phenotype really early. Already at 6 weeks of age, we have this fibrosis developed in the liver. So we can start to treat these mice already when they are 6 weeks of age. We have a chronic inflammation, which is preceding this fibrosis. So it's this chronic inflammation that ends up damaging the organ. And this is what you see in many human conditions. So it mimics the human conditions in that sense. And the best thing in my mind is that we have this 100% reproducibility. And this means that if you look at fibrosis in the livers of NIF mice at the same age, you get the same picture in all of the mice. And this means that when you treat the mice and you see an effect on the fibrosis, you know that this effect is actually due to my drug and not due to variances in levels to start with. So this model, the NIF mouse, is out on the market as a liver fibrosis model. We have signed 8 customer contracts so far. We have customers in Europe, we have customers in Australia and in the US. Only this year we signed 4 contracts, and we predict that we will sign at least 1 or 2 more before the end of the year. And when I say we, I mean basically these 4 people. So InfiCure consists of 4 people. They are working actively in the company. So it's me, myself, I'm the CEO. I have a PhD in human genetics. I have more than 15 years of experience from research, project management, from my positions at Umeå University, Copenhagen University, and my time in San Diego at La Jolla Institute for Allergy and Immunology. Then we have my co-founder, Dan Holmberg. He's a professor in immunology, but he's also the CSO of the company. He has more than 35 years of experience from research and working with project management, experimental animals, and so forth. He's been working at several universities, both in Sweden and in Denmark, and now he's working part-time as a CSO and part-time as professor of immunology in Lund, at Lund University. And then we have the 2 most important persons, actually, Maria Hörnberg and Julia Nilsson, because Maria and Julia are the ones that are doing the actual work on the mice. They are treating the mice, and they are doing all the analysis. Maria has a PhD in molecular biology. And she's our lab manager. She has a long experience from working with experimental animals. She used to be the coordinator at the transgene facility in Umeå. And Julia, she's a research scientist. She's been working for a long time with this NIF mouse from the time when we had it in the academia, actually. So it's the 4 of us that are working in the company. But besides the 4 of us, we have, of course, a board of directors. So we have our chairman, Lena, who is sitting over here, Lena Söderström. She has more than 30 years of experience from managing positions at several companies. We have Mattias Eriksson, who is an investment manager at Partner Invest, but he has a background in life science since he's been working at AstraZeneca and at Pfizer. And then we have Dan as well, but I already introduced him. So what will happen then in the future for InfiCure? Well, we will actually do an RNA sequencing, which might not say a lot to all of you, but we're going to do this. We're going to look at the gene expression in these mice, since we have a lot of potential customers that ask us, so is my target of interest expressed in this mouse model? So we're going to do this RNA sequencing to be able to actually answer these questions and get more contracts. Then we are also validating the NIF mouse as a kidney fibrosis model, because as I told you, it's a spontaneous model. We see the fibrosis in the liver, but we do also see it in the kidneys. So the liver is validated. Now we are going to work on the kidneys, or we are working on the kidneys. And we estimate that we'll actually have the NIF mouse on the market as a kidney fibrosis model in mid next year. But that's not it. This NIF mouse and a variation of this NIF mouse, we have some preliminary results saying that these 2 models could actually be models for NASH, non-alcoholic steatohepatitis. So we are validating that as well, and we estimate that we will have them on the market in the end of next year. But that's not it. We're also going to, as I said, we have customers in the US, in Australia, and in EU. but we're going to look at the Korean market, because we are lucky enough to have a project with Business Sweden where they're actually going to help us to get into the Korean market and see what we can do for them there. Okay, my last slide is gone, apparently, but I don't know, must be missing. Anyway, we are fighting fibrosis. That was basically what I was going to say, and thank you very much. And if you're in the fibrosis field, or if you know somebody who's in the fibrosis field, keep us in mind. Thank you.